All right, so you want the need to know on-sample ionization for mass spec, and that's exactly what we're here for. And as always, I have with me,
Right.
well, an expert in the field. So mass spec is like this amazing molecular detective, right? It helps us figure out what stuff's made of, how much of it there is, and it's used everywhere. Chemistry, biology, forensics, you name it.
Absolutely, yeah. Can't overstate how versatile it is. But before we can get to the detective work, we got to prep the evidence, right? That's where ionization comes in, giving these molecules a little electric boost.
Okay, so it's like the molecules need a backstage pass to even get into the mass spec party.
Exact.
And it looks like there's a ton of ways to do this, each with its own strengths and weaknesses, I guess. So for this deep dive, let's break down some of these ionization methods, and we'll use the material you sent over as our guide.
Sounds good to me.
Perfect. Okay, so let's start with the OG, the classic, electron ionization, or EI. Can you give us a quick refresher on how that works?
Sure, basically, we hit the sample molecules with a beam of high-energy electrons. About 70 electron volts, if you want to get specific. And that's enough energy to knock an electron off the molecule, creating a positively charged ion,
Hmm.
which is what the mass spec needs to do its thing.
So like molecular billiards, but with electrons as the cue ball.
Yeah, that's a good way to put it.
Okay, cool. Now, in your notes, you mentioned needing a library to make sense of EI data. What's that about?
So one of the great things about EI is that it produces these very predictable fragmentation patterns. When the molecule breaks apart, it does so in a consistent way. And over the years, scientists have built up these huge databases,
Yeah.
like the NIST one, containing the EI spectra of thousands and thousands of compounds.
It's like a fingerprint database, but for molecules.
Yeah,
So
exactly.
you match the fragments from your undone sample to what's in the database, and bam, you've got an ID.
Exactly.
That's super cool.
Yeah.
But I'm guessing there are limitations to EI, right? Not every molecule wants to play electron billiards, I'd imagine.
Right. Not every molecule is a good fit for EI. One of the big things is that it works best with volatile compounds, stuff that can easily turn into a gas. And also, those high-energy electrons, they can cause a lot of fragmentation, which can make the spectra pretty complex, sometimes hard to interpret.
So what if you have a molecule that's a little more on the fragile side? What do you do then?
In those cases, we might use something a little gentler, chemical ionization or CI. Instead of blasting the molecules with electrons, we use a reagent gas like methane or ammonia. This gas creates ions, and then those ions react with our sample molecules in a much softer way.
Okay, so it's like a molecular handshake instead of a full-on collision.
Yeah, that's a good analogy.
Now, you specifically mentioned needing to keep an eye out for the parent ion in CI. What's so special about this parent ion?
So the parent ion, also called the molecular ion, that represents the intact molecule before any fragmentation happens. And because CI is gentler, it produces less fragmentation. So that parent ion is often much easier to see in the spectrum.
Okay, makes sense. So it's like CI helps those shy molecules get on stage without getting overwhelmed.
Exactly.
Okay, now in your notes, you also mentioned atmospheric pressure ionization, or API. What makes these techniques different?
Well, the main thing is right there in the name. They work at atmospheric pressure, which means they're a lot easier to pair with liquid chromatography or HPLC.
Right, HPLC, that's like the workhorse for separating mixtures. So
Yeah.
being able to connect HPLC with mass spec directly, that's huge.
It really is, opens up a lot of possibilities.
And you said one of the most common API techniques is APCI, right? Atmospheric pressure chemical ionization.
Yeah.
So is that basically CI, but just happening at atmospheric pressure?
Pretty much. It's great for ionizing a wide range of molecules,
Yeah?
especially smaller ones and nonpolar ones. It's also pretty tolerant of salts, which can be a problem for other methods.
Yeah, thinking about my work with pharmaceuticals, that salt tolerance is a big deal.
For sure.
So APCI works well with HPLC, handles salts. Sounds like a winner for analyzing complex drug mixtures. Now, you also mentioned APPI, atmospheric pressure photoionization. Is that similar to APCI?
It is. It's also an API technique, but instead of a corona discharge like an APCI, we use UV light to ionize the molecules.
Ah, so we're giving them a little UV tan to energize them.
You could say that. It works really well for molecules that have low ionization energies, meaning they're easy to kick an electron off of, and sometimes we even add a dupant to help things along.
Okay, so a little extra boost to get them ready for their mass spec debut.
Exactly.
Now, I know we should keep moving, but this is really interesting. So are there setups where you can combine APCI and APPI? It seems like they'd complement each other pretty well.
Yeah, you're right. There are combined sources, and they can be really powerful. But maybe let's save that deep dive for another time.
All right, fair enough. But there's one more technique I want to talk about, one that's really changed how we look at large molecules, electrospray ionization, or ESI. It's amazing how it can handle those big, fragile molecules like proteins without tearing them apart.
Yeah, ESI has been a game changer for sure.
So how does it work? What's the magic behind ESI?
Well, it starts with an electric field. We apply a high voltage to a liquid sample, creating a very fine spray of charged droplets. And as those droplets shrink because the solvent is evaporating, the charges on their surface get closer and closer together. And that leads to some interesting stuff.
Like the Taylor cone you mentioned earlier.
Yes,
Yeah.
exactly. So the Taylor cone, it's this cone-shaped protrusion that forms at the tip of the emitter when the electric field is just right. It's really important because it focuses a spray and helps produce these tiny, highly charged droplets.
So it's like a molecular fountain sculpted by electricity.
A good way to visualize it.
Okay, so we've got these charged droplets, but how do we actually get to the individual ions we need for the mass spec?
So as the solvent keeps evaporating, the droplets get smaller and smaller, and the repulsion between those charges on the surface, it gets stronger and stronger, until eventually the droplet just can't hold it anymore, and it explodes in a process called Coulombic fission, releasing those ions into the gas phase.
Whoa, so it's like a microscopic fireworks show?
In a way, yeah.
That's so cool. And I've heard that ESI is really good at generating multiply charged ions.
It is.
Is that related to the molecule size or weight?
Yes, it is. One of the really cool things about ESI is that the number of charges an ion picks up, it's directly proportional to its mass. So bigger molecules will tend to have more charges. And this is super helpful for accurately figuring out the masses of large molecules.
Like a built-in molecular scale.
Yeah, pretty much.
That's so clever. But I've also heard that ESI can be a little finicky or sensitive to certain things.
That's true. There are a lot of factors that can affect how well it works. The solvent is a big one. For example, the solvent's surface tension plays a role in how much voltage you need to start the spray. And different solvents can affect the stability and size of those charged droplets, which impacts the ionization process.
So it seems like every detail matters in ESI.
It does. There's a lot to consider when you're setting up an ESI experiment. Things like flow rate, solvent composition, even the shape of the emitter.
I can imagine.
But all that control, it's part of what makes ESI so powerful and versatile. We could probably spend a whole deep dive just on ESI optimization.
That's probably true. But for now, let's move on to another technique I'm curious about. Inductively Coupled Plasma Mass Spectrometry, or ICP-MS. You mentioned it's the heavy metal rocker of ionization methods.
Yeah, I did.
So what makes it so special?
Well, unlike the other techniques we've talked about, ICP-MS doesn't use electrons, chemicals, or even UV light to ionize the sample. It uses a plasma, super hot plasma,
Okay, how are we talking?
up to 10,000 degrees Celsius.
Wow, that's hotter than the surface of the sun.
It is.
So what happens to the sample in that kind of heat?
It gets completely atomized and ionized, broken down to its basic elements with all the electrons stripped away. This makes ICP-MS incredibly sensitive. We can detect elements down to parts per trillion.
That's like finding a single grain of sand in an Olympic-sized swimming pool.
It's pretty impressive.
It is, and you said we can analyze multiple elements simultaneously, right?
We can.
So it's really useful for fields like environmental monitoring, geochemistry, food safety.
Yeah, all of those and more.
So it's basically an essential tool for understanding the elemental makeup of, well, everything.
Pretty much.
Now, I know there's a whole other category of ionization methods, the desorption ionization techniques.
Right.
These are the ones that use energetic particles to, like, kick ions off a surface.
Yeah, like tiny cannons shooting out ions.
Okay, so one of the earlier techniques in this category is fast atom bombardment, or FAB. I have to admit, that name always makes me think of fashion magazines. But I'm guessing it's not quite as glamorous.
Not quite, no. So in FAB, we use a beam of fast atoms, usually xenon or argon, to bombard the sample. Now, the sample is mixed with this thick liquid called a matrix.
Oh, the matrix. We've seen that before with MALDI.
Right. It comes up a lot in these ionization techniques.
So what's its role in FAB?
It's really important. The matrix absorbs most of the energy from those fast atoms, which protects the sample molecules from getting damaged. And as the matrix absorbs that energy, it vaporizes, carrying the sample molecules with it into the gas phase where they can be ionized.
So it's like a molecular launching pad.
Yeah, a good way to think about it.
So FAB is good for molecules that don't like being vaporized directly.
Exactly. It's particularly useful for non-volatile compounds and thermally unstable ones, things that would break down under heat. It's also pretty sensitive, which is always a good thing.
Sounds great. But are there any drawbacks to FAB?
Well, one thing is it's not very good at separating mixtures. The spectra can get crowded, especially if you have a complex sample, and because it relies on that matrix, it's not ideal for volatile compounds, things that might evaporate before they can be ionized.
Okay, so it's not perfect for every situation.
Yeah.
But it definitely has its strengths. So what came after FAB in the world of desorptionization?
Well, there's secondary ion mass spectrometry, or SIMS. It's similar to FAB in that it uses a beam of energetic particles to knock ions off a surface. But instead of using atoms, SIMS uses ions.
So it's like a more focused beam, using ions instead of atoms.
Yeah, that's a good way to put it.
And what's really cool is that SIMS can give you spatial information about the sample surface.
Right, we can map out where different molecules are located, which is incredibly useful for things like material science and nanotechnology.
So now we're talking about high-resolution imaging.
Exactly.
I can see how that would be super valuable for understanding how materials are put together at the nanoscale.
For sure. And the technology keeps getting better. Some instruments combine SIMS with a time-of-flight mass analyzer, or TOF SIMS, which gives you even better resolution and sensitivity. And you mentioned in your notes that you're interested in instruments that combine TOF SIMS with an Orbitrap mass analyzer.
I am. They sound incredible.
Yeah, they are pretty impressive. The Orbitrap is a very high-resolution mass analyzer. It can give you incredibly accurate mass measurements. So combining it with TOF SIMS gives you the best of both worlds.
That's amazing. It's really amazing how these techniques keep evolving, pushing the boundaries of what's possible. Now, I have to ask, are there any desorptionization techniques that can handle really large molecules, like massive protein complexes?
There are. One technique that's really pushing the limits is massive cluster impact, or MCI. It's like FAB in SIMS, but instead of single atoms or ions, it uses these huge clusters of molecules to bombard the sample.
Well, how big are we talking?
We're talking clusters that can contain tens of thousands of atoms, with masses up to 10 to the 8th atomic mass units.
That's like throwing a molecular boulder at the sample.
Pretty much.
So what happens when these massive clusters hit the surface?
They transfer a huge amount of energy to the sample, causing desorption and ionization of even very large molecules.
It's incredible how we can manipulate matter at such a tiny scale, using these molecular wrecking balls to reveal what's hidden inside.
It is pretty amazing, and MCI has been really useful for analyzing large biomolecules and polymers, things that were really hard to study with other methods.
Sounds like a powerful tool.
Yeah.
Now, I know we've been focusing on those newer desorption ionization techniques, but there are a couple of older ones that I want to touch on. LDI and MALDI.
Ah, yes, the classics.
So LDI, laser desorption ionization. That was one of the first techniques that used a laser to desorb and ionize molecules from a surface. But it works best for small molecules, right?
Yeah, LDI can be tricky for larger, more delicate molecules.
So size matters in LDI,
It does.
and that's where MALDI comes in. Matrix-Assisted Laser Desorption Ionization.
Right.
MALDI is famous for analyzing big biomolecules, like proteins and DNA.
Exactly.
So how does it work?
Just like with FAB, we mix the sample with a special matrix. This matrix absorbs the laser energy, and when the laser hits the sample, the matrix explodes, carrying the aniline molecules with it, and boom, you have ions.
It's like a microscopic rocket launch.
Yeah, kind of.
And because the matrix absorbs most of the energy, it's a lot gentler on those big, delicate biomolecules.
Exactly. That's one of the key advantages of MALDI. It can handle large, fragile molecules that other techniques would destroy. And it's very sensitive. You only need a tiny amount of sample.
So it's fast, efficient, and gentle. What's not to love? But you mentioned earlier that there's an art to choosing the right matrix for MALDI.
There is. The matrix is really important for absorbing that laser energy and transferring it to the analyte molecules. But different matrices have different properties. So finding the right one for your specific sample and application, it can take some trial and error.
I can imagine. But once you get it right, you can do some pretty amazing things with MALDI.
You can.
I've read about some really cool applications in proteomics and imaging.
Yeah, MALDI is super versatile. We can even couple it with ultra-high-resolution mass analyzers like FTICRMS, which gives you incredible mass accuracy and resolution.
That sounds incredibly powerful.
It is, and it shows you just how powerful and versatile mass spectrometry can be when you combine these different ionization techniques with advanced mass analyzers.
This has been fantastic. We've covered so much, from electron beams to lasers and those matrix-assisted techniques. I'm already feeling a lot more knowledgeable, and I bet you are too, listening in.
Me too.
Mm,
It's amazing how many different ways there are to ionize molecules,
right.
and each one has its own strengths and weaknesses. So choosing the right one for the job is really important.
Absolutely. It's all about finding the right tool for the question you're trying to answer. But before we wrap up this part of our deep dive, there's one more ionization technique I'm curious about. Plasma desorption, or PD. It's an older technique, but it played a big role in how mass spec developed.
Oh yeah, PD, that's an interesting one.
So what's the story with PD?
Well, this one's a little different. It actually uses a radioactive source to generate ions.
Radioactive material. That sounds a little dangerous.
It's all very carefully controlled. The source, usually californium-252, undergoes spontaneous fission, releasing high-energy fission fragments.
So we're talking about harnessing the power of nuclear fission to create ions.
You could say that.
Wow.
So these fission fragments hit the sample, which is on a thin foil, causing desorption and ionization of the molecules.
So it's like a microscopic nuclear explosion.
In a way, yeah.
Oh, that's wild.
It is pretty wild. But PD was actually really useful for analyzing large non-volatile molecules back before techniques like MALDI became widely available.
It's fascinating to see how far mass spec has come, from radioactive sources to lasers and everything in between.
It is a lot of ingenuity has gone into developing these techniques.
That is for sure. All right, well, let's take a break and recharge our ionization batteries. We'll be back soon with even more insights and a few more mind-blowing analogies, I'm sure.
Looking forward to it.
All right, so ready to jump back into this ionization whirlwind. We've covered so much already, but there's still so much to explore.
Absolutely. It's amazing how many different ways there are to nudge these molecules into the mass spec.
It is, right?
Yeah.
And you've been great at explaining how each method has its own little personality, its strengths and weaknesses, when to use it, when maybe not to.
It's all about finding the right tool for the job, just like anything else.
Exactly. So what else have you got for us? What other ionization tricks are hiding up your sleeve?
Well, how about we start with a technique that might sound a bit familiar. Thermospray ionization, or TSI.
TSI.
It's a bit older, but it was really important for developing what we call LCMS, liquid chromatography mass spectrometry.
Right. LCMS, the dynamic duo of analytical chemistry, separating and identifying all in one go.
Exactly. And TSI was one of the first techniques that let us connect those two.
Connect HPLC with mass spec, right?
Yep.
So tell me more about TSI. The name kind of suggests there's some heat involved, right?
You got it. In TSI, we dissolve the sample in a volatile solvent, and then we heat it up as it flows through a capillary tube. This creates a spray of droplets, and as the solvent evaporates, the analyte molecules are left behind carrying a charge.
So there's like a tiny molecular sauna getting those molecules all prepped for the mass spec.
I like that, a molecular sauna.
But you mentioned in your notes that TSI has some limitations, right?
Yeah, it can be a little tricky to get it working just right. And the sensitivity isn't always the best. It can also have trouble with certain types of molecules, like those that are really nonpolar or break down easily with heat.
Okay, so maybe not the most popular technique these days, but still an important part of mass spec history.
Definitely.
Now, what about field desorption, or FD? You mentioned that one too.
Yes, FD. This is another technique that uses strong electric fields to ionize molecules. It was actually one of the first that could analyze non-volatile thermally labile compounds.
Non-volatile thermally labile, meaning they don't like heat, right?
Exactly.
Because it's gentle on those fragile molecules. But how does it actually work?
Okay, so picture this. We have a special emitter, and it's covered in these tiny carbon whiskers called dendrites.
Carbon whiskers, that sounds kind of cute.
They do, right. So we apply a high voltage to these dendrites, creating a really strong electric field at their tips. And when we put the sample on the emitter, the electric field is so strong that it pulls electrons off the molecules, causing ionization.
So it's like a molecular tug of war, but the electric field always wins.
That's a good way to put it.
And because you're not using heat or anything, FD is very gentle. Right?
Very gentle, produces very little fragmentation.
Sounds like the perfect technique for those delicate molecules that need a little extra care.
It can be. But of course, there are limitations. FD can be a bit tricky to operate, and it's not as sensitive as some of the other techniques we've talked about.
Right. There's always a trade-off, isn't there? Sensitivity, gentleness, ease of use.
It's true, but luckily we have this whole arsenal of ionization methods to choose from, so we can usually find the best one for the job.
That's the beauty of mass spec, right? So many options. Okay, so in your research, you highlighted glow discharge ionization, or GDI. What makes GDI stand out?
GDI is really good for analyzing solid samples directly. You don't need much sample prep, which is a big advantage.
Direct analysis, less prep work. Okay, I'm listening.
So in GDI, we create a plasma by applying a high voltage between two electrodes. And we do this in a low pressure environment.
Plasma, again, we talked about that with ICP-MS, right? It's this superheated, partially ionized gas.
Right. So in GDI, we're basically creating a mini plasma torch to ionize the sample.
And that sputters ions off the surface of the solid sample.
Exactly.
Sounds very efficient.
It is. And GDIs have been really valuable in fields like materials, science, surface analysis, and thin film characterization.
Cool. Now, switching gears a bit, you also mentioned field ionization, or FI.
Yes, FI.
Another one that uses strong electric fields.
Yes, it's similar to FD in that it uses a very sharp tip to create a strong electric field. Usually the tip is made of tungsten. But instead of those carbon whiskers we talked about with FD, FI relies on the sharpness of the tip itself to concentrate the field.
So it's like a tiny lightning rod, but for molecules.
Exactly. And when a molecule gets close enough to the tip, the electric field is so strong that it can actually pull an electron away through a process called tunneling.
Tunneling electrons. That sounds like some serious quantum physics.
It is, and because it's so gentle, FI is often used to study the structures of complex molecules, or to analyze those short-lived molecules that form during a chemical reaction.
Wow, capturing those fleeting moments in a reaction? That's incredible.
It is pretty amazing.
Okay, so let's talk about a technique that seems to be getting a lot of attention these days. Desorption, electrospray ionization, or DAI.
Ah, DII, a very cool technique.
It combines desorption and electrospray ionization, right?
And it does.
Well, what are the benefits of combining those two approaches?
Well, one big advantage is that DSI lets us analyze samples directly in their natural environment. We don't need any sample preparation.
No sample prep. That sounds very convenient.
It is. So in DSI, we have a spray of charged solvent droplets, just like in ESI. But instead of spraying those droplets into the mass spectrometer, we aim them at the surface of the sample.
So it's like you're washing the sample with those charged droplets.
Yeah, that's a good way to visualize it. Those droplets hit the surface, pick up analyte molecules, create secondary ions, and those ions are then pulled into the mass spectrometer.
A molecular car wash for mass spec analysis.
I like that.
And DSI is versatile, right? You can use it for all sorts of things.
Absolutely. It's used for imaging, analyzing surfaces, studying biological samples, all without having to extract or purify them.
That's really impressive.
It is a powerful technique.
Okay, now I know we're running out of time for this segment, but there's one more technique I want to touch on. Surface-Assisted Laser Desorption Ionization, or SALDI.
Ah, yes, SALDI, another laser desorption technique.
It's similar to MALDI, but with a little twist, right?
Right, so with MALDI, remember we use a matrix to absorb the laser energy. But in SALDI, we use a special surface to help with the desorption and ionization.
So the surface itself is the key player.
Exactly.
What kind of surfaces are we talking about?
Well, there are a few different materials that work well. Things like porous silicon, graphite, even nanoparticles. The important thing is that the surface absorbs the laser energy efficiently and helps release ions from the sample without causing too much fragmentation.
Okay, so we're basically giving the laser a helping hand.
Exactly.
So what are the benefits of using a surface instead of matrix?
One advantage is that SALDI can be gentler than MALDI, which makes it good for analyzing very fragile biomolecules. It can also be helpful for analyzing samples that are difficult to dissolve in a matrix, like some polymers and inorganic materials.
Sounds like SALDI has a lot to offer. But are there any downsides to using a surface?
There can be. Sometimes the surface itself can interfere with the analysis, or it might not be compatible with certain types of samples. And choosing the right surface for a specific experiment can take some trial and error.
Right, finding the perfect balance, it's always a challenge in mass spec.
It is, but that's what makes it so interesting.
Well said. This has been fantastic so far. We've learned about so many different techniques, from the classics to those pushing the limits of what's possible. I can't wait to see what's in store for us in the final part of our deep dive.
Me too. We've got a few more ionization techniques to explore, and then we can wrap up with some thoughts about the future of ionization and what might be coming next.
Sounds good to me. Stay tuned, everyone, for the grand finale of our ionization extravaganza. Okay, so we're back for the final part of our ionization journey, and it's been quite a ride learning about all these different techniques.
It has been. We've covered a lot of ground, haven't we? Electrons, lasers, plasmas, even a touch of nuclear physics.
It's amazing how creative scientists have been in finding ways to give those molecules a little electric nudge.
Absolutely. And as we've discussed, each method has its own, let's say, personality, strengths, weaknesses, ideal applications.
Exactly. So for this last part, what else do you have in store for us? What other ionization secrets are you going to reveal?
Well, how about we start with a technique that takes a rather cool approach? Cold spray ionization, or CSI.
CSI, like crime scene investigation. Are we going to solve a molecular murder mystery?
Not quite, although it does help us analyze some pretty delicate samples.
Okay, so tell me more about this CSI.
So in CSI, we actually cool the spray down to very low temperatures before it enters the mass spectrometer,
How cold are we talking?
typically below -20 degrees Celsius.
Wow, that's chilly. Why the need for such a frosty reception?
It's all about protecting those fragile biomolecules. You see, many of them, like proteins and peptides, are very sensitive to heat. They can break apart or change their structure if they get too warm.
Ah, so it's like giving them a cryogenic spa treatment to keep them relaxed and intact.
Exactly. By cooling the spray, we can gently ionize these molecules without damaging them, allowing us to see their true form.
Okay, so how does it actually work? Do we literally freeze the sample?
Not exactly. We dissolve the sample in a volatile solvent, just like in ESI, and then we use a nebulizer to create a fine mist of droplets.
Okay, and then?
Then we cool that mist down to those super low temperatures before it goes into the mass spectrometer.
So it's like a gentle, icy breeze carrying those molecules into the mass spec.
Exactly. As those cooled droplets travel, the solvent evaporates, and the analyte molecules are left behind carrying a charge. And because everything is kept so cold, those fragile biomolecules stay happy and intact.
I can see why this is a popular choice for analyzing proteins and other sensitive biomolecules.
It is. Now let's switch gears a bit and talk about a technique known for its speed. Sonic spray ionization, or SSI.
Sonic spray, that sounds fast.
It is. We use a high-velocity gas flow to create the spray.
Okay, walk me through it.
So we use a nebulizer to create a fine mist of droplets, similar to ESI. But instead of an electric field, we use a high-velocity gas flow to charge those droplets.
So it's like a gust of wind sweeping those molecules into the mass spec.
Yeah, that's a good way to put it. That gas flow helps to evaporate the solvent and create charged ions. And because it's such a fast process, SSI is great for analyzing lots of samples quickly.
Ah, I see. That would be super helpful in areas like drug discovery or environmental monitoring, where speed is key.
Absolutely. And SSI is pretty versatile. It can handle a wide range of compounds from small molecules to larger biomolecules.
So it's like the all-terrain vehicle of ionization methods.
I like that analogy. Okay, ready for one more?
Absolutely, bring it on.
All right, this one's really cool, cutting edge stuff. Laser ablation, electrospray ionization, or LASI.
LAESI. Okay, that sounds pretty high-tech. Lasers and electrospray. What's going on here?
So it's a two-step process. First, we use a mid-infrared laser to ablate or vaporize a tiny bit of the sample.
So we're basically using a laser beam as a microscopic scalpel, carefully removing a sample.
You got it. And this creates a little plume of material containing the analyte molecules.
Okay, and then what?
Then we aim an electrospray plume at that ablation plume, capturing those analyte molecules and ionizing them.
It's like a laser-guided electrospray capture mission. So we use the laser to target exactly where we want to sample from, and then the electrospray swoops in and collects those molecules.
Precisely. And the amazing thing about LASI is that you can analyze samples directly without any preparation. Tissues, plants, even single cells, all in their natural state.
Wow, that's incredible. No sample prep. That's a game changer.
It is. And because we're using a laser, we have incredible spatial resolution. We can target very specific areas.
It's like having a molecular microscope, letting us see the molecular makeup of anything.
Exactly. And that brings us to the end of our ionization journey.
Wow, what a trip it's been. I feel like I have a whole new understanding of mass spectrometry and how important ionization is for unlocking those molecular secrets.
Me too. And the really exciting thing is that this field is constantly evolving. New techniques are being developed all the time, pushing the limits even further.
It's amazing to think about all the discoveries that are still waiting to be made, all thanks to these incredible ionization methods.
It is. And on that note, we'd like to leave you with a final thought. What if we could combine the best features of all these different techniques into one ultimate ionization method? A method that's gentle and efficient, can handle all sorts of molecules, and gives us amazing sensitivity and spatial resolution.
That's a challenge worth thinking about. It would be like having a Swiss Army knife of ionization techniques ready to tackle any molecular mystery.
Exactly. And who knows, maybe you, listening out there, will be the one to invent it.
Thanks for joining us on this deep dive into the world of ionization.
It's been a pleasure exploring this fascinating field with you. Until next time, keep those scientific minds curious and those mass specs humming.